Hi, I want to show you our new software grandR, which is for the analysis of nucleotide conversion RNA-seq data. In general, nucleotide conversion RNA-seq, such as SLAM-seq, is a powerful method for comprehending highly dynamic gene expression changes, but the analysis of this data remains challenging. Also, there is a lack of computational tools for doing this. This is why we developed grandR. But first of all, let me introduce you to SLAM-seq. And SLAM-seq cells get treated with a nucleoside analog, for example, 4SU, for some labeling time. 4SU then gets incorporated into newly synthesized RNA instead of uridine. After chemical conversion, it gets converted into a cytosine or cytosine analog. Those T2C mismatches can then be observed in the reads, telling us which RNA is new and which is old. Unfortunately, this whole process is not perfect and relies highly on the 4SU incorporation rate. In order to get unbiased estimates of the amount of new and old RNA, our lab previously developed a software called grandSLAM, which uses a binomial mixture model to give us the new to total ratio and the posterior distribution of it, which we can then take to further downstream analyze. It's important to know that labeling over extended periods of time or with high concentrations of 4SU can affect cell viability or RNA metabolism. And it's important to control for such effects. In grandR, this can be done by comparing 4SU treated with equivalent 4SU naive control samples. If we look at a PCA of those samples and we see a segregation, this already indicates that there has been an effect. Another way of looking at this effect would be by comparing the log-fold change of 4SU treated to 4SU naive samples to the NTR rank. If we see here a correlation like here in the right-hand plot, then there has been an effect. If it looks like on the left-hand side, everything's fine. If we see something like this, we can then decide if we need to exclude the sample or if we could potentially rescue it. grandR is a big toolbox and also contains functions for many other things. For example, differential analysis. And here we introduced a new technique for estimating degradation and synthesis rates from snapshot data. Previously, the log-fold changes of new and old RNA have been used to get any information about synthesis or stability. For synthesis, these also correlate very well, but for the half-life, the log-fold change of RNA does not correlate with the log-fold change of the half-life. By using our statistical approach, we see that our estimates are more accurate for the synthesis and even more accurate for the half-life. Additionally, we compared our approach by using a ROPE, so region of practical equivalence, with DSeq2 to see if we can accurately identify changes in synthesis or half-life. And what we found is that for synthesis, ROPE and DSeq2 showed very similar sensitivity and specificity, but for the half-life, our approach showed a higher sensitivity and specificity because DSeq2 also detected changes in both and not only in half-life. Also, we have several methods implemented in grandR for estimating kinetics. For example, linear regression, non-linear least square regression, another R package, so pulsar, which was made for a slightly different type of data. And lastly, we also use the NTR that we get from grandSLAM. And what we did here is we simulated data and we compared the true half-life to the estimated half-life, and we saw that the NTR and NNLS approach were the most accurate ones. It's also important to note that 4SU is not immediately available for transcription. It first needs to be transported and preprocessed. So, for example, for the 1 hour time point, effective labeling could, for example, be only half an hour because in the first half hour of labeling, there was not enough 4SU to effectively label. And we actually see this in data. So, for example, here we see that this time point would fit rather here. And we can also see this effect globally. So for the first time point, the global residues were mostly negative. We can use a joint maximum likelihood approach and calibrate this. And we see that after calibration, this effect was completely rescued. Also, grandR contains many functions for visualization that can be accessed either programmatically or over a Shiny-based web interface. In the Shiny-based web interface, we have mainly two tabs. The first is the gene level, the second is the global level. On the gene level, we have our analysis table that we can search, filter, and scroll through. We can select the gene and then get gene level plots, which are completely customizable and interchangeable. Also, we can download everything basically. In the second tab, we have the global level plots where we can select genes that will then appear in this box. We can add a highlight or set a highlight that then will be set for all global level plots. From this global level plots, we can identify genes that we then want to have a closer look at here and vice versa. In general, this tool is really, really handy when working with collaboration partners for sharing the results and also for intuitively analyzing them. In summary, grandR streamlines the analysis of nucleotide conversion RNA-seq data by offering easy-to-use functions from quality control up to publication-ready figures.